Hello, welcome to the genetics video notes. The standard that we are working on is SC.7.L.16.2. Determine the probabilities of genotype and phenotype combinations using Punnett squares and pedigrees. Let's get into it. What are Punnett squares? Good question. Punnett squares are models that allow people to predict the genotype, which is the genetic code, and the phenotype, or the physical trait of offspring from a cross. A cross is what we call a mating between two parents. If you look in the picture here, you will see that the cross is between two heterozygous parents. The two parents are heterozygous. They are both big A, little a, and big A, little a. And then we see the completed Punnett square right here. What are the steps to making a Punnett square? Great question. First, we have to establish the cross or the breeding that is being evaluated because we have to know the genotypes of the parents. Then we have to draw our Punnett square, set it up with the parents' genotypes, and then we'll do our stop, drop, and roll procedure to fill in the Punnett square. Before we do anything else, we must establish the cross. We need to know what the genotypes of the parents are, and we need to know what trait we are evaluating. For example, we can evaluate the trait of ear shape. And in our fictional example here, the dominant trait is rounded ears, the recessive trait is pointy ears. The dad and the mom both have rounded ears, and they are heterozygous. Remember, heterozygous is referring to a genotype that has two different alleles. So the genotype is one dominant allele, represented by a capital letter, and one recessive allele, represented by a lowercase letter. When we write the cross, we are always going to write it father's genotype, an X, and the mother's genotype. I have to pick some alleles to represent the dominant and recessive traits, so I'm just going to pick the letter E because ear starts with E. It's random. The letters we use as alleles are like variables in math. They have the meaning we assign to them. In this case, I'm assigning big E as the dominant ear shape, which is rounded, and little e as the recessive trait, which is a pointed ear. Now I have to draw my Punnett square. So first I'll draw a box. And then I have to draw a line to split the box in half horizontally. Remember, horizontal means side to side. And then I'm going to draw a line to split my box in half vertically, which means up and down. Next, I'm going to set up the Punnett square. Remember, our cross is big E, little e for the male, crossed with big E, little e for the female. The father's alleles are written first, then the mother's when we write the cross. The father's alleles are written on top of the Punnett square, not inside the boxes, but above the boxes. The mother's alleles are written to the left of the Punnett square, not inside the boxes, but outside of the boxes. So the father's alleles are going to be arranged like this. The big E is going to be over the left-hand column. The father's little e is arranged over the right-hand column. The mother's big E is arranged to the left of the top row. The mother's little e is arranged to the left of the bottom row. Now that our Punnett square is set up, we're going to stop, drop, and roll. So stop. Look at your Punnett square. Is it set up correctly? In this case, yes, our Punnett square is set up correctly. Now we're going to drop the father's alleles into the boxes below them. So we're going to write the father's alleles into both of the boxes that are directly below each allele. So dad's big E is going to drop down into the first box, and it will drop down into the second box. Dad's little e will drop down into the first box. Dad's little e will also drop down into that second box. Notice how I wrote a little e in the column to the right, and I made sure the little e was off to the right side of the box. Remember, we always write the dominant allele first. It's a good indicator to you as to what the phenotype will be. When you see the dominant letter first, it helps you to see what the phenotype will be. It will be the dominant phenotype. Now we have to roll the mother's alleles across to the right. We will write the mother's alleles in both boxes directly to the right of the allele. So mom's big E is going to roll over to the first box. Mom's big E is going to roll over to the second box on the top row. Mom's little e is going to roll over to the first box on the bottom row. And mom's little e will also roll over to the second box on the bottom row. This is your completed Punnett square. This is accurate. This is complete. Let's see that again in color. In an effort to make sure you understand how to arrange the letters in a Punnett square, I'm showing you the arrangement again. And I've changed the colors of the letters so that you can track where they're moving a little more easily. So again, we got to stop, make sure the Punnett square is set up correctly. The Punnett square is set up correctly. We're going to drop the father's alleles and write them in both boxes directly below the allele, directly below that letter. So dad's big E is going to drop down to the first box. Dad's big E is going to drop down to the second box. Dad's little e, which is in red, is going to drop down to the first box. Dad's little e in red will also drop down to the second box. Now we have to roll. Mom's big E will roll to the first box, and it will continue rolling over to the second box. Now mom's little e, which is in green, will roll to the first box, and mom's little e in green will roll to the second box. Now we have another complete Punnett square. Look very carefully where the black E is placed, the black big E, which is one of the father's alleles, drop down to the boxes directly below it. The dad's little e, which is in red, drop down directly into the boxes below it. The mother's big E, which is in blue, rolled right to the boxes directly beside it. The mom's little e at the bottom, on the bottom row, rolled to the right into both boxes. Let's see that again. Remix. So I'm going to show this to you one more time, and I'm going to explain it a little bit differently. Maybe this will help you understand better. I want you to recall that you get half of your genetics from your mother and half of your genetics from your father. When you look at a genotype that has two letters, you know that one of those letters, which represent our alleles, came from your mother, and one of those letters, which represents your alleles, came from your father. So the point of the Punnett square is to show how the different alleles can be distributed in a particular mating or breeding, okay? So remember that meiosis is the process that produces gametes. When gametes are produced in males, only one allele for each gene is going to be in that sperm. So when you look at the father's big E, that represents a sperm that has the dominant allele. When you see the father's little e, you see it represents a sperm that has the recessive allele for ear shape in our hypothetical, mythical, fictional example here. For the mother, she's going to produce some gametes. One of her eggs might have the big E for the dominant allele. She could also have eggs that have a little e for the recessive allele. When a breeding occurs, it's random. It's completely random. We don't know which egg and which sperm containing which alleles are going to unite to create a new life. So we are representing all of the possible combinations. So what possible combinations are there? Well, in this case, dad's big E could match up with mom's big E. So now we have an offspring that is homozygous dominant. Or dad's big E could match up with mom's little e. And then we have a heterozygous offspring. Or dad's little e could match up with mom's big E, which gives us the possibility of another heterozygous offspring. Or dad's little e could match up with mom's little e. And we have the possibility of having an offspring that is homozygous recessive. Remember, these are just possibilities. What it means is if you had a large enough group of offspring and you looked at their genetics and their phenotypes, then you would see a rough distribution that is similar to the Punnett square. That means if the pointy ears and the rounded ears are on baby mice, and you have 100 baby mice that are bred over the course of a couple of years, the rough distribution of genotypes would be about 25% of the offspring would have the homozygous dominant genotype. Approximately 50% of the offspring would have the heterozygous genotype, and approximately 25% of the offspring would have the homozygous recessive genotype. That is the information we get from a Punnett square. It does not guarantee that offspring will occur. It just tells you the probability that offspring with a certain genotype or phenotype will be produced. So let's dig a little deeper into that, shall we? A Punnett square has 4 boxes in it. Each box is a genotype possibility, and it represents a 25% chance of a specific genotype occurring. So remember, 4 quarters make $1. Each quarter is worth 25 cents. When we look at the Punnett square with its 4 boxes, we know that each box is going to represent 25% of the total. All 4 boxes represent 100% of the total possible outcomes of the cross that we are looking at. So let's evaluate this Punnett square right here. How many of the offspring are homozygous recessive? Well, we take a look at the Punnett square here. We remember homozygous recessive means there are 2 recessive alleles in the genotype. One box has the 2 recessive alleles, so 25% of the offspring are homozygous recessive. How many have the dominant phenotype? Well, remember my example that if LeBron is in the gym, nobody will notice Miss Roebuck. If the dominant allele is in the genotype, you will not notice the recessive allele in the phenotype. So 3 of these boxes have the dominant allele. That means that you will see the dominant phenotype in those offspring. What about how many are heterozygous? That would be 50%. Remember, heterozygous means there are 2 different alleles. So the offspring have a dominant allele and a recessive allele. There is a 50% chance that that is going to happen. And what about a recessive phenotype? There is a 25% chance of a recessive phenotype. Only one box has the homozygous recessive genotype, which is the only way that we can see the recessive phenotype in the offspring. Remember, if LeBron is in the gym, you will not notice Miss Roebuck. If the dominant allele is in the genotype, you will not notice the recessive allele. If the dominant allele is in the genotype, you will not notice the recessive allele. If LeBron, the tough, strong, excellent basketball player, is in the gym doing slam dunks and beating everybody at the game and giving away checks for $1 million, nobody will notice Miss Roebuck, who has the athletic ability of a banana peel. That is how genetics work. If the dominant allele is in the genotype, you will not notice the recessive allele. The only way to see the recessive phenotype is if you have a homozygous recessive genotype. All right, let's move on to pedigrees. So a pedigree is going to show a family tree and it will track a certain trait. So these can tell you how a trait is passed down through generations. If you get really good at pedigrees, you can tell the if the particular trait is autosomal dominant, autosomal recessive, sex linked. You can tell all of these different things when you get really good at Punnett squares. At pedigrees. Pedigrees are super simple. There are 4 symbols that you need to know that will tell you whether or not a member of the family is affected by the trait. Affected just means that the family member has the trait. So whatever the trait is, it could be hairy knuckles or it could be male pattern baldness. If the shape is shaded in, that means that the family member has the trait. So there are 2 symbols that represent male or female family members. Males are represented by a square. A white square is going to represent an unaffected male. That means the male does not have the trait. A shaded in or colored in square is going to represent an affected male. That means he has the trait. Females are represented by a circle. A white circle or an unshaded circle represents an unaffected female. She does not have the trait that you're looking for or that you're tracking in this pedigree. A shaded in or colored in circle means the female is affected by whatever this trait is. When I try to remember the difference between male and female for a pedigree, I think women get pregnant, and when female people get pregnant, they have round bellies. When a lady gets pregnant, she's got a round belly. A pregnant person has a round belly. The belly is round. So we're talking about a female. And the males are squares. There's only 2 options. So if the circle represents the round belly of a female, the square must be a male. All right? Maybe that'll help you remember what the symbols mean. We have a pedigree here, a very simple pedigree, and it shows us what the different symbols that are used to connect the pedigree are for. So the marriage line is a horizontal line that connects married people, or it can show where a mating occurred. This can be in unmarried people with kids. It could be, if you're doing a really complex pedigree, maybe a plant or an animal. We also have the line of descent that is going straight down, and it shows that there were some offspring. Then we have the sibling line. Notice how the line of descent goes straight down, and then there's another horizontal line. The horizontal line here to show siblings has a line, however many siblings there will be a line going straight down to connect the sibling to the parents. Notice how there is not a straight line between the siblings. A straight line between the siblings would show that they are married or had offspring. And that is atypical. Do not do a straight line between the siblings. The siblings must be connected through this symbol right here. The siblings are directly connected to the marriage line, not each other. Then we have the generation number. So there are two generations in this pedigree. Generation 1 is the mom and the dad. Generation 2, there's a son and a daughter. In this pedigree, nobody is affected by whatever trait they were tracking. This is just a pedigree of a family that does not have the traits. All right, this is a big pedigree and it has a lot of information. I can tell you with some certainty that the trait that they're tracking, let's say it was, let's just say male pattern baldness. Let's say this is male pattern baldness. I think that the trait that we're tracking is recessive. And I'll tell you the evidence that I have that this is recessive. If you look in generation 1, person 1 and person 2 are unaffected, but they have offspring in generation 2 that is affected. If you look in generation 2, person 1 and person 2 are unaffected, and they have 2 affected children. If you look in generation 3, persons 7 and 8 are unaffected, but they have 2 offspring in generation 4 that are affected. And in generation 4, person 1 and 2 are unaffected, but they have offspring in generation 5 that are affected. Remember that it is possible for the parents to have one phenotype, but pass a different phenotype off to their offspring because the parents are heterozygous. So the parents are both, let's just pick a letter to be our alleles. Let's just say R. So both parents are big R, little r, and 2 of their offspring inherited at least one big R, which is why they show the dominant phenotype. But one of these offspring inherited little r from mom and little r from dad, which is why they have this trait, even though the parents do not have this trait. All right, let's take a look at these pedigrees. Lord Spindletune is a poodle. Let's see his pedigree or his family's pedigree. So Lord Spindletune is a male poodle. He got loose and had puppies with a female mutt. There were a total of 6 puppies born. There were 3 males, 3 females. And our little story here says that 2 of the females and 1 male have curly fur like Lord Spindletune, and the other 2 males and the other female have straight fur. We are tracking curly fur. So when you look at the pedigree, we see the male, Lord Spindletune, the female, which was a mutt. We don't know her name. And we see there's 6 offspring. 2 females have the trait. 1 male has the trait of curly fur, just like the dad. As you can see, my drawing is not perfect, but you can definitely tell the squares versus the circles. And they're shaded in, and I added my generation markers. So this is perfect. This is absolutely acceptable as a pedigree. Next, we have Wally and Jenna's family. So here's Jenna and Wally. Now, when I read this, I saw that there were 3 generations, so I made sure I drew a little bit higher up. So Jenna and Wally have 2 children, a boy and a girl. Their boy child has been married, and their boy child has children. And we have our 3 generations here. Wally and Jenna are the first generation. And Julio, the son who got married, his kids are in generation 3. Now we're tracking red green color blindness. So all of the males have red green color blindness. And one of Julio's daughters also has red green color blindness. So Wally and Julio's son and one of Julio's daughters all get shaded in because they have the trait for color blindness. And we have our generation markers set up. Perfect. Last but not least, we have the Olufemi family. So in this family, they have the trait of mind reading. So we see grandpa and grandma have 5 kids. They have 2 sons and they have 3 daughters. And our story here tells us that the mind reading skips a generation. So none of grandpa's kids have mind reading. But the daughter got married and her son is also a mind reader. And since we're tracking the trait of mind reading, we're going to shade in the grandpa who has mind reading and the grandson who has mind reading. We added our generation markers. Boom! Another perfect pedigree.